Hello and welcome to this edition of the Seneca webinar, where Melanie Wesoff from BBMRI-ERIC will share ethics ELSI considerations from fair to fair data sharing. My name is Marta Llorell Linares, and I'm involved in the Seneca project on behalf of Findability. I will be hosting the webinar today. I would like to start by making you aware that this webinar is being recorded, and that includes the question and answers session at the end. So we have reserved some time at the end of the webinar where the audience can ask questions. If you have any, please write them in the question box of the GoToWebinar application, as you see on this slide. And you can already write the questions before the presentation is finished or at the end, as you prefer. I'll introduce you the Seneca project. Seneca stands for Common Infrastructure for National Cohorts in Europe, Canada, and Africa, and it's a 4-year project funded by the European Union's Horizon 2020 program and the Canadian Institute of Health Research. Seneca's vision is to accelerate disease research and improve health by facilitating transcontinental human data exchange. To achieve this, Seneca is working on several challenges. The first one is federated data discovery. So how, as a researcher, you find cohort data of interest? And this could be for a specific phenotype or genotype, for example. And once you find the data, you will need permission to access it. So Seneca is working on an interoperable authentication and authorization infrastructure. Once you get access to the data and you want to ask research questions across cohorts, there's the need for a harmonized cohort-level metadata. The ultimate aim is to enable federated data analysis for research and healthcare applications. So Seneca is working with a set of demonstrators across cohorts in both clinical and research settings. And on top of all these challenges, there's a fundamental need for a transnational harmonized ELSI framework to address ethical, legal, and societal issues. Seneca has also an outreach and training program to increase awareness and build capacity in these areas. The webinar today is part of the event How Fair Are You?, which consists of a webinar series and a hackathon to provide background knowledge and practical advice in order to facilitate the uptake of FAIR principles. We have already run 5 webinars, and if you want to watch the recordings of those webinars, you can find them in the Seneca website and YouTube channels. On the 28th and 29th of April, we'll run a hackathon, a practical online event where we'll work on the implementation of FAIR principles in 3 parallel streams: cohort data, software, and training materials. And on the 28th of April, there will be a webinar entitled Application of FAIR Principles in the Seneca Project. You can register to the webinar independent of the hackathon. So if you want to know more about this and register, please visit the Seneca website. Today's presenter, Dr. Melanie Wesoff, is a social scientist and research officer at the European Research Infrastructure for Biobanking, BBMRI-ERIC. She is also lecturer at the Department of Science and Technology Studies at the University of Vienna. She studied sociology at the University of Vienna and at Royal Holloway University of London, and completed the postgraduate program Sociology of Social Practice at the Institute for Advanced Studies in Vienna. Melanie holds a doctoral degree from the University of Vienna. Her dissertation was awarded the prize for best thesis in 2017 by the Research Network Gender and Agency of the University of Vienna. At BBMRI-ERIC, she is involved in various H2020 projects, where she focuses on the ethical and societal implications in biobanking, data sharing, and artificial intelligence, as well as governance and stakeholder engagement. She is also leading the International Task Force Societal Issues at BBMRI-ERIC. At this point, I will hand over to Melanie.
So today's topic is about the ethics and ELSI considerations when it comes to FAIR and FAIR. So it will not be so much about, the technical aspects of the FAIR principles, but considerations that comes with it, more on the side of FAIR or fairness. So while this whole series is about the FAIR principles, so why do we need ELSI here, or what is ELSI at all? Just a brief reminder. So ELSI stands for Ethical, Legal, and Societal Issues, and it covers very broad areas of research here. And this means, for instance, legal requirements related to data privacy and data protection that somehow also overlaps with some ethical challenges connected to it, especially in regard to consent models or protecting participants' autonomy. And also challenges coming with new technologies, also connected to the secondary use of samples and data, and return of results and data sharing. Which also then, when talking about participants and their involvement, also includes some social or societal aspects, in particular public engagement, and also recognizing patients as active participants in research, and also involving them at an early stage of the establishment process of a project, also in view to for the sustainability of these projects, or biobanking in particular, where I'm concerned with mostly within BBMRI-ERIC. So considering all these issues are very important in doing research. And as ELSI, as you see, covers many broad, wide areas, there is, well, no such thing as the ELSI expert, because that would be a person being all, a lawyer, an ethics person, a social scientist, a philosopher, and so on and so forth. So as you have seen from the nice introduction by Martha, my background is in social science, so I will talk basically about the ethical and societal issues. So talking about ELSI and all the issues you might concern with might bring you to the following challenges or questions. So can we use or reuse data without informed consent because they are anonymized? I will address this question in today's talk. This goes together with questions, what if the data protection regulations change? So also the time frame here, and how to protect data in the future, even if you don't know exactly what future research will be able to uncover, to do. And if this is not known, can participants withdraw their data from projects, from platforms? And given the technical developments, is anonymization still a valid concept in the age of genetics and big data, especially when it comes to data linkage? And also, who is in charge? So what about power relations and social justice? So who is included, who's excluded, who has the power to do what with which kind of data? And also giving also the kind of data that is produced also in secondary uses. So what about incidental or unsolicited findings? Should participants be informed about it? And what happens if participants became patients because of that kind of information? What care mechanisms are in place? And as I mentioned before, for sustainability, it's also important to ask the question how participants, publics, patients can be involved in all the research or research practice we are going to do. And what about collaborations with health industry? So just a couple of questions that are of relevance for now, also concerning existing data sets and future uses in research. And these questions are not as abstract as they perhaps appear on this slide. If we just think about how to improve informed consent, what does anonymity mean in times of genomic research? What about the role participants or patients or all the people who donated data and sample to research, let me just take you a little bit back in time, and especially to a very prominent case of what research is able to do. So basically for research, a success story, but it also shows the relevance of thinking about social justice and the role of the participants. This slide gives an overview of what's possible with the HeLa cell line. I know it's quite small. I will walk you a little bit through this slide. So the HeLa cell line, when we start here in 1951, oh, my mouse does not shut up, was generated from Henrietta Lack's tumor sample taken without her consent. Henrietta Lack was a Black American woman and was treated for cancer in Johns Hopkins Hospital. In the lab, her cells turned out to have an extraordinary capacity to survive and reproduce, and the researchers shared them widely with other scientists. And being the first immortalized human cell line, these cells became of importance for science and technology, and they have been at the forefront of many biomedical developments in the 20th century, which you can see here on these slides. For example, in virology, for the polio development of the polio vaccines, they were used to also establish kind of or build a supply industry for biomedical research. They were used for many other applications for infectious diseases. They even went to space, and given on that, it was kind of really groundbreaking, the research that was possible based on these cells, and they continue to be relevant for research. So if you are interested in this story, especially in the life of Henrietta Lack and how this story unfolds, I will highly recommend the book by Rebecca Skloot, The Immortal Life of Henrietta Lack. Today, work done with the HeLa cells underpins much of modern medicines. And as I said, they have been involved in really key discoveries in many fields, including cancer, immunology, and infectious diseases. And one of their most recent applications has been in research for vaccines against COVID-19. So HeLa cells were everywhere, so also in many, many labs, and it was discovered that the HeLa cell line has been contaminating other used cell lines. And this led scientists to search for genetic markers to distinguish the original HeLa from other cell lines. So in 1973 and 1974, the Flex family have been contacted by scientists to collect blood samples from family members, again without consent, and for what is a cancer test. So even if you would say, okay, obtaining consent for research participants or donors, whatever, was not a common practice in 1951, especially also for a Black woman. It was the same case in the 1970s, where these blood samples were taken from family members. And this is also highly controversial, especially as a result of the colonial past and the striking contrast between the Lack family's economic status and the biotechnology giants who have established business around the HeLa cell line. So what does this have to do with today's topic of fair and fair? So let us turn to a more recent chapter in the HeLa story. Yeah, so which dates back to 2013, when a research group at EMBL published an article in which the authors provided the HeLa genome as supplementary data. And such genome sequences of different organisms, including the human genomes, and being a product of international consortia working to produce a common research for the research community, were produced and were producing data according to what we would call now the FAIR principles. And that is why the publications initially was seen as a very positive effect for the scientific community, providing this kind of information or the genome sequence of the HeLa cell line. And the wide sharing of data was promoted, for example, in open access policies. Coming with that was a press release by EMBL, where the question appeared, can we infer anything about Henrietta Lacks or her descendants from this sequencing? And the short answer was no, we cannot infer anything about Henrietta's genome or of her descendants from the data generated in the studies. Well, not everyone shared this opinion, especially Janif Ehrlich, who is known for, who has proven that it is possible to use online public databases to find identities of people whose anonymous DNA sample has been sequenced and published online. And his reaction on Twitter was, I think, quite clear. So he answered to the question of, can we infer anything about Henrietta Lacks from the HeLa cell line? And the answer was no. And he just commented it with nice lie EMBL. Also, Jonathan Eisen, a biology professor at the University of California, Davis, tweeted, a bit stunned that the people publishing HeLa genome appear not to have gotten consent from the family. Emphasizing here the important topic of consent. And also, Rebecca Skloot wrote in the New York Times, as I mentioned, she's the author of the book of Henrietta Lacks and the HeLa cell line. And she said in this article that the publication of the HeLa genome without consent isn't an example of a few researchers making a mistake. The whole system allows it. Everyone involved followed standard practices. They presented their research at conferences and in peer-reviewed journals. No one questions about consent. And this opens up a lot of questions, also, which I'm trying to cover a little in this presentation. So, especially when it comes to anonymization or anonymity and genomic re-identifiability, consent, research practice, and benefit sharing. So, looking back at how these samples were originally obtained and how the data or the sequence is used now, I think shows that this case is still really important, and it also points us to that life and research today are high datafied. We live in a datafied world, and in medical research is increasingly becoming a data-intensive activity in which health-related data, genomic and other data are being collected, reused, and linked on a large scale. So the question is now, while we see all the potential this sharing could have and all the success the use of this data could have, how we can share ethically. Before I open up this question, I just want to let you know, if you are interested, we are currently working on a paper with my colleague Kaya Akyus and other members of the BBMRI-ELSI family, where we work on more details around this case I was just mentioning. And we gave a webinar last year called Looking Back, Going Forward, about the story of Henrietta Lacks. You can find it on the BBMRI YouTube channel. So coming back to the question about how to share data, how to share it ethically. So the approach what we find in the literature is consent or anonymize, and this hints to a normative link between consent as research ethics principle and consent as a legal basis, and also to the close link between anonymization and privacy. So quick reminder on consent forms or informed consent forms. There is not just one, there is a whole spectrum going from specific consent, which means that you have to repeat the full process of informed consent for each research project. And the other end would be kind of blanket consent that does not restrict uses. What we see in biobanking research, or the research we mostly talk about, is that broad consent became the model for informed consent. And broad consent allows the collection and use of samples and data at the time of enrollment for ethically approved biomedical research. And samples stored can be used for new studies that fall within the scope of the original broad consent without reobtaining consent from participants. So quickly, the definition of broad consent. And this use or this practice of broad consent is justified with a minimal risk for participants and the benefits for the public good, and also the impossibility to obtain consent for every project that is done. So a major critique on broad consent is raised on the fact that participants are asked to consent to the use of their donations in yet unknown future research projects, questioning whether this type of consent could be understood as based on informed decision-making. So the risk related to the future use and sharing of health data in various projects across various countries have raised concerns about the potential harms. And more detailed, I just listed a couple of critical points here. It was discussed if broad consent, I mean, it could imply that kind of the ethics work is concluded when consent is obtained. So if you have the consent from the participants, you're done with ethics. But the question is, ethics should be seen as an ongoing process. As I mentioned before, things are changing. The future is not known now exactly what we are able to do with samples and data. So the question here is it meaningful or even legally valid, understood as specific, explicit, freely given and informed, that consent is obtained at a one-off event at the time of data collection and then allowing various uses of it. And participants are asked to consent to future research projects, making data available for linkage, reuse, and analysis for yet unknown future purposes. So also the consequences are unknowable at the time data is collected or aggregated. So can we speak of really properly informed? So is it informed decision making at all if you cannot give this kind of information? And if the question that is attached to that is consent fails to provide potential participants with enough information, so can they then even withdraw their consent? Can participants imagine what's happened? What could be done in future studies? Even we as researchers do not know exactly. And what are future risks? So what could properly inform mean? I just want to mention two other cases here. The one is the Havasupai case of a tribe, the Havasupai tribe, which gave blood samples for a genetic research into type 2 diabetes. The consent says it's research on the causes on behavioral or medical disorders, but samples were also used for other questions, including ethical migration, metabolic disorders, alcoholism, and schizophrenia, the latter a stigmatizing condition within the tribe. And participants said later that they were never aware of having given consent to these questions that go beyond the original research. So what is the definition of informed consent here, and especially if you include vulnerable groups? Another example is the use of data that have been collected by UK Biobank with broad consent that allows health-related research. And these data were used and they were reviewed by and approved, or the use were approved by the UK Biobank Access Subcommittee, and they were used in a study that was on kind of the genetic background or on same-sex sexual behavior. So information or data that was donated for health-related research purposes was used in the study about same-sex sexual behavior, and the question was now if for the participants, if they would also see this kind of question included in health-related research. And it also opens up another question, as for instance, homosexuality has long been removed from disease classification, and making such a claim by including it into health-related research could be normatively problematic, as it is implying that it has to do with a disease. That's just one aspect of which we cover in a more recent paper we published, where we asked the question of the role of the social into big data research, or research questions that should be studied with the use of big data sets. So properly informed, does it mean I have to anticipate every kind of information or uses or everything and include it in the consent forms? We may end up here, that we kind of create these monsters of consent forms and a kind of transparency paradox. And then what also could happen is that we hand over all this responsibility and everything we actually don't know over to the participants. So this could also not be the best solution here. And talking of participants, what do they want? And even if they don't know exactly about the research that is going on, what could happen, participants want to have a choice. And previous studies have come to the conclusion that reconsent is preferred when the circumstances under which this research has taken place change. And most of the research participants want to have a say how the samples and data are used. For instance, I quoted here 3 studies saying that it became a little bit problematic when genomic data and health information is shared among many actors, including many countries. I think this is a recent study, and also a very important question now about sharing about genomic data. So the trust in kind of this sharing practice is relatively low. A study that I was conducting with a colleague, which was based on an interactive setting where we have focus groups with members of the public and participants and patients. And here we see that there is an understanding that there is a kind of appropriate research practice, and the decisions on giving consent is based on these understandings, and also linked to understandings how data are used currently. So as I said, we live in a datafied world. People are aware that there could be misuse or multiple use of such data. And also important is the use of broad consent in low and middle income countries, which should be used using safeguards and really engagement with the community. So in order to avoid exploitation. So the term freely given consent somehow implies then that participants should at least have choice. And so that was the side of the participants. Let's have a look at what, for instance, Biobankers are saying about sharing practices and what should be covered in informed consent procedures. And then we see quite high numbers that sharing data and samples with non-commercial research partners or commercial research partners, in particular health industry partners, as well as other countries, that this kind of information should be included in informed consent procedures. So going back to the question about how to do that now, how to handle it. If one-off consenting is not enough, if you cannot include every or anticipate everything that all participants should know, there are many other kind of consent models. One you might heard of is the idea of a dynamic consent, basically not in that sense a research model, but a digital decision support tool, so a platform, and it means that it is personalized, that is a communication platform which allows participants to manage their preferences continuously. And there also have been the discussions if participants are even interested to do that over and over again. But that's then a matter of choice, at least to have the choice and the decision to do that if they want to. So it basically means that there is an initial kind of a consent procedure, and but within that, the consent decisions could be specific. So the benefits are that it connects the researchers and the participants and increases engagement, and also making recontact easier. So it could also be the case that research participants could be interesting candidates for other kinds of studies. But there are, of course, also risks to that. So that it kind of could fail to protect the autonomy of the participants, as it and to ensure this high level of information and understandings, and that consent is then obtained in kind of an uncontrolled conditions, and also the matter of a digital divide. So not everyone is able to participate in this kind of consent. Okay, so if you say, the consent, this is very complicated, and if we just use data, we can anonymize them. As I mentioned before, in the healer case, you see that the case of genomic re-identifiability, I mean, is important to consider now. And the question is now, what do we mean by anonymization or re-identification? So anonymization was frequently seen as the minimum requirement necessary to protect data subjects' privacy, and even despite the possibility of re-identification through cross-referencing with other kinds of data. And genomic data is highly distinguishable. With only 30 kind of SNPs, the individual can be identified, as I mentioned before, Janif Ehrlich, who has showed that it's actually, if you know how to do it, it's not so difficult. And the risk of matching anonymized genetic data with data from other data sets involving research environments, and increases the possibility of re-identification. So complete anonymization is becoming increasingly illusory as this possibility of cross-matching large data sets improves. And it's also not about the consent with individuals, or not only individuals are concerned anymore. So, but the concept of anonymization is also problematic, as it represents consent as a concept relevant only to the identifiable individual, whereas group-level harms from analysis of aggregated data are clearly possible. And big data operates on groups of anonymized individuals, and while this could have matter a little for the individual, it could for the group. So discrimination and stigmatization is therefore a real risk. Yeah, so coming to the question about this kind of re-identifiability and also to re-identify ethically, especially in terms when it comes to incidental findings. And the already difficult question, should we go back to the data subject or to the donor of samples and let them know. So there is already an intense discussion about whether to provide feedback on individual genetic results from genome sequencing to research participants, as this could have consequences for health decisions making. And if we anonymize all the data and kind of cut the link, then we are not able to go back to the data subjects. So here is the balance quite clear about what FAIR wants, also that the loss of data granularity, which is actually needed, and FAIR points to having rich data sets to enable reuse and broad consent would then kind of not make sense if we minimize the data so much. So, or for instance, the identification is not even feasible because it's simply too expensive or not doable for other reasons. And it could also mean a loss of valuable medical information, and therefore also the opposite of FAIR. So bringing that together, so from the kind of tissue issue in how to work or doing research with samples in a specific studies, we are now in a more scenario of a data issue, because the risk profile has changed. So as I mentioned before, the risk that was kind of included in the broad consent considerations, that is relatively low because the samples are not used particularly for a specific kind, but in the course of other treatments. We have now the risk of informational harm, either in form of lags or hacks, or risk of re-identifiability and stigmatization in connection to genomic analysis. And also the understanding what qualifies health research data is likely to change over time. But if we see consent as a one-off kind of procedure, then participants gave consent on what it means for them at the time they gave consent. So there is a situatedness of consent based on societal values, definition of what health disease is, privacy, and so on and so forth. So yeah, many things to consider. So, okay, is there a way out? Well, the GDPR also has the research exemption. And because consent might be to obtaining consent will be challenging for in many cases. So the basis of public interest in the field of public health and the scientific research allows for data subject consent to be circumvented. And this is a controversial question, whether this is ethically acceptable to use instead of consent. And this could also open up that these uses are also could also be used by private or commercial entities. And appropriate safeguards needs to be guaranteed, even if it's there is the option to circumvent consent. It still guarantees the right to inform of the data subject and the principles of fairness and transparency. And despite the lack of consent, processes to inform data subjects still have to be in place. And as I mentioned before, and the critique of this one-off consenting based on really changing environments was also to consider like research, also consent more as consenting, so more as a process. And I quoted here a suggestion for an ethical research exemption instead of consent. And here you see it kind of outlined how this procedure could look like, including these procedural safeguards. And they would include technical and organizational measures, such as strongly encouraging anonymization and pseudonymization where possible, having a data protection officer in place, also data protection impact assessments, and also transparency in the decision-making process. So you can see it's ongoing here, and also outlines that multiple actors or responsibility is also shared, and there are multiple actors in place to take care of all these safeguards and take care if the research proposed participants, for the recent participants have consented to the data to be used, is still in line with what they were consenting. So this brings us also to another question of fair and equitable use of data, especially in connections to vulnerable groups or low and middle income countries, as it's the case with Seneca, as also Africa is included here. And as already mentioned, there is a long history of unethical use of African data and material, as well as potential repeated misuse and commercialization of African genetic material. And equitable and ethical use of data, as well as equal partnerships with African countries for fair data sharing of data and return of benefits among all involved stakeholders is vital for good governance. So, for instance, the H3 Africa Guidelines for Consent note the importance of a quote, robust governance framework that should seek to promote global health and research equity and take into account 5 key elements: respect, authentic community engagement and trust building, the preservation of privacy and confidentiality, feedback of results, and capacity strengthening. And overall, public participant and community engagement is inevitable here. So again, the process is an act also of balancing de-identification, anonymization on the one hand, on the other hand, ethical re-identification in case of incidental findings. And what we've also seen was the data minimization, which is included in the GDPR on the one hand, on the other hand, rich data to enable reuse, which is what the FAIR principles actually include. And also the question of the level of granularity needed on the one hand, and on the other hand, the granularity that could be harmful as it reveals too much information, for instance, in the case of vulnerable or ethnic groups and so on. I mean, there was just a few considerations when we came to fair within fair data sharing, which is not only about the technical requirements, but also including the ethics and societal and legal aspects. Finally, I just want to point out that if you have also questions, you can also refer to the BBMRI-ERIC Services, especially to the ELSI Help Desk and the ELSI Knowledge Base, where you can find some documents, or just get in touch with the ELSI Help Desk. And I stop now for the moment. If you want to get in in touch, just send me an email. And yeah, thank you for the opportunity to giving this presentation today.
Thank you very much, Melanie, for the presentation. So if you have any questions, you can write them in the question box in the GoToWebinar application. We have already one, Melanie, so I'll read it out for you. Thanks for a great overview, Melanie. For me, the big pink elephant in the room remains the disparity between how Silicon Valley treats personal rights to data protection and how it's treated in health research. Should researchers be more entrepreneurial and point out the benefits of research and risk-reward ratios relative to government and corporate data uses of personal information?
Okay, a big question. Yeah, I mean, I guess what the question is pointing to is the use of data for different kinds of reasons, and if we kind of limit or regulate uses that we perhaps don't want, or which are there is a controversy when it comes to Facebook and all the other kind of data uses, or even direct to consumer testing services, like for instance 23andMe, and they are, or this kind of companies, yes, they are really successful in obtaining samples and especially data, while I think research, or for instance biobanking, which I'm more connected to somehow, have to stick to really clear regulations and doing all these kind of consent procedures more according to the law. So is there fairness here? I'm not sure if the answer is that public institutions or biomedical research should act more as these companies. I think that's a question more about how to communicate with participants, which kind of information you provide, because these companies, where they are really successful, is making it kind of an yeah, a pleasure to visit their websites and the kind of information you retrieve from them. So this would also be a question in how to return results or insights into research. And I think, yeah, I just can make the point that the engagement with participants is really needed here, not to be better in advertising it, but better in engaging with participants and clearly communicate why this is used and useful. I think there are many success stories out there why this kind of data sharing has helped actually many people. So I don't know if this answers the questions, but good engagement strategy, good communication strategies are really important, so that people know to what they are consenting to. And this is not just risky, but it also enables a lot of research that helps many people.
Okay, thank you, Melanie. So we just received a question that is a bit of follow-up on that one. Can you provide some examples of successful engagement activities? Where do they begin and when do they end?
Yeah, thank you for this question. As I mentioned, all these things should be more thought of a process than kind of one-off activity, consent obtained. That's it for the participants, to put it very short. I think what I was hoping to make you aware, that all these questions and considerations are around, and the trust of research participants is really important. And examples for engagement are starting very early. So if you yourself are planning kind of a project or working in these environments and are planning a study, you really need to think of these engagement activities, which starts from information, but engagement is more than just one directional information campaigns. Early on, this could be there are very different options. Mainly it's about how information is provided, either on websites, but also who's there to answer questions, and also like events. I also were included in a couple of, doing focus groups, involving them in discussions about what is going on, even also invite people to your research facilities. For instance, I also participated in one where we walked through a biobank just to have an idea on it and just meet people who are working there. Often it's really to who's there, who are the humans there, who is in charge of data protection. Who's there to ask, even for you as a researcher, if you start such a project. Familiarize yourself with your own kind of institutions or also possibilities. So who is the data protection officer? Where do you get all this kind of information? And also plan resources for such activities also in your research projects. So not just postpone it until we'll solve that later, or we will have the consent questions and the anonymity considerations later. Really plan that. Because as I said, this is a process, and this process also needs resources. So providing these informations in a certain way, opening up, answering all these questions, that's also a matter of resources. Which, you should also plan in handling this kind of data, and this should already be included early on in your research projects. So I guess that's perhaps what I can say now. There are good practice examples out there in Europe, but what they do have in common is that they really started early on with communicating, inviting participants, showing who's there, that it's just not an anonymized repository of biobanks somewhere, but there is a real interest in getting in touch.
Thank you, Melanie. So we have another question that says, thank you for a great presentation. Can you please elaborate a bit more why context matters so much?
Okay. I already covered a lot of this in examples of a lot of aspects that comes with the use of data. Sorry, could you again repeat the question?
Yeah. Can you please elaborate a bit more why context
matters
so much?
Yeah. Because as I said, you will get in touch with all these kinds of questions, and what we also see from other examples where context mattered was when it comes that something was not in line with public expectations or something went wrong. Often research projects are conducted also on public resources, so there is a responsibility here how to use these resources. And we also saw some examples, for instance, when it comes to Biobank, where public support were lost because of some practices, because of not considering some things, and this is really also a risk for these kind of projects or also infrastructures, because not even that there is a responsibility on your side as researchers, it's also if publics or patients especially also are not good involved, or also with their concerns, because basically it's not that there are research participants which of course do not understand every aspect of your research or are not interested or whatever, but they, as I mentioned, they also live in a world that's highly datafied. And before, the question was also pointing to some companies in Silicon Valley, which where is critique about data uses, and this also somehow reflects to many other uses of data. So you should be aware that people in general are also living in a datafied world and become more aware of how the data is used, and also have questions, and also the societal issues are really important, just to avoid kind of discrimination and the representation, and to grant the representation of certain groups, just in view of also social justice. And this is important, of course, for the research, but also when it comes to these questions of engagement and how people are represented there, because if you lose the public support, then you're really at risk in also when it comes to could be a risk for research project or the funding of research projects.
Thank you, Melanie. We have a couple more questions, but I think I'll make only one more because of the time. So it's, why do you use the plural in publics?
Kind of social science terminology. Thank you for pointing that out. There is not one big group of the public out there. There's always the distinction into different groups. So when you think of public, of whom do you think, who could you reach? So is it a highly educated group to which you have in mind, or who you communicate with? Is it a certain other big group? So it's not kind of precise enough, or does not represent the kind of diversity of different groups in the public if you keep it not that way. But there are multiple publics in that sense, that you also could speak to different kind of groups within a society, given on, I don't know, educational backgrounds, different age groups when it comes to media. So there are very different distinctions of how who would reach whom, and who you would include when you think of the public or publics.
Thank you for the explanation. Can you go to the last slide just to have a reminder of the next webinar? Yes, so thank you very much, everybody, for attending this webinar. So the next Sineca webinar will be on the 28th of April on application of FAIR principles in the Sineca project. And you can register if you go to the Sineca website and find the event. So thank you very much again, and see you next time.